Hi, my name is Hannah, and welcome to my scientific communication assessment. I hope you enjoy. Hi. So this presentation is all going to be about ghrelin, which is sometimes called the hunger hormone because it stimulates appetite, increases food intake, and fat storage. So ghrelin is predominantly produced in the stomach and interacts with stomach ghrelin as well as hypothalamic growth hormone releasing hormone and somatostatin in the regulation of the pituitary growth hormone secretion. What regulates ghrelin? The most known factor for the regulation of ghrelin secretion is feeding. Plasma ghrelin concentration increases when fasting and decreases after food intake. Plasma ghrelin concentration is low in obese people and high in lean people. Exogenous growth hormone decreases stomach ghrelin, mRNA expression and plasma ghrelin concentration, but does not affect stomach ghrelin stores. Ghrelin is also identified in the hypothalamus. Ghrelin-containing neurons are found in the arcuate nucleus of the hypothalamus, a region involved in appetite regulation. The orexigenic effect of hypothalamic ghrelin is regulated through a neuronal network involving food intake. So ghrelin is secreted primarily from the stomach in response to hunger and starvation. It circulates in the blood and serves as a peripheral signal informing the central nervous system via the vagus nerve to stimulate feeding. With all this said, ghrelin interacts with the GI system and the endocrine system, but also the circulatory systems. Okay, so the site of action and cell signaling. Ghrelin is a gastric peptide hormone discovered as being the endogenous ligand of growth hormone secretagogue receptor. Ghrelin is a 28 amino acid peptide presenting a unique N-octanylation. Ghrelin is mainly produced by a subset of stomach cells and also by the hypothalamus, the pituitary, and other tissues. Transcriptional, translational, and post-translational processes generate ghrelin and ghrelin-related peptides. Ghrelin is the only known peptide hormone modified by a fatty acid. Ghrelin is synthesized by endocrine XA-like cells in the fundus mucosa, representing about 20% of gastric mucosal cells in humans. Ghrelin is the natural ligand for the growth hormone secretagogue receptor, GHS or GHSR. Ghrelin is predominantly expressed in the digestive system, with highest expression levels in the gastric mucosa. Gastric mucosa is composed of 5 endocrine cells: endochromaffin cells, endochromaffin-like cells, D cells, G cells, and XA-like cells, respectively secrete serotonin, histamine, somatostatin, gastrin, GABA, and ghrelin. In the gastrointestinal tract, ghrelin expression gradually decreases from the duodenum in the colon. Circulating ghrelin originates in vast majority from gastric mucosa and gastrointestinal tract. In the pancreas, ghrelin immunoreactivity colocalized with glucogen-producing cells, insulin-producing cells, a new type of endocrine cells, and ANA cells. So GHSR1 signaling. Coupling of the GHSR1A G protein involves a third intracellular loop. The lack of the third intracellular loop in the GHSRB prohibits it from coupling to G proteins. GHSR1A activation leads to a subsequent activation of phospholipase C, inositol triphosphates, and intracellular calcium pathways. GHSR1B signaling is unable to bind to acyl or deacyl ghrelin. GHSR1B, considered in the past to be functionally inactive, is now believed to act as an important modulator in ghrelin-induced GHSR1A signaling. Okay, so now I'm going to talk about a disease state to do with ghrelin. I've chosen Prader-Willi syndrome. PWS is associated with failure to thrive in infancy and progressive hyperphagia and obesity in childhood. This progressive weight gain is associated with hyperghrelin anemia and increased insulin sensitivity. PWS is a genetic syndrome that results from lack of expression of paternal genes located on chromosomes 15, Q11 to Q13. In the newborn period, children with PWS have hypotonia, poor suck, decreased arousal and failure to thrive, and often require tube feedings for several weeks to months. By 1 to 6 years of age, such patients develop insatiable appetite, progressive weight gain, short stature, hypogonadism, cognitive and motor delayed development, behavioral difficulties, and sleep disturbances. Adolescents and adults with PWS have high circulating concentrations of ghrelin. Hyperghrelin anemia might contribute to the hyperphagia and excessive weight gain in PWS. The number of oxytocin neurons, the putative satiety neurons in the hypothalamic paraventricular nucleus is marked decreased in PWS. This is presumed to be the basis of the insatiable hunger and obesity of patients with the syndrome. So their decreased neurons in the hypothalamic nucleus is the reason why their hunger is much more and the obesity is more prevalent with this syndrome. Okay, so I've just got a reference list here of everything that I use throughout the presentation. Also, this includes everything that I said is included. Okay, so I've just got a reference list here of everything that I use throughout the presentation. Also, this includes everything that I said is included.